Okay, thank you everyone to attend this online conference. So it's great to share with you about the comprehensive omics analysis to explore the application in clinical oncology. So that's the experience from BGI. So I think first of all, we can have a brief overview of the whole picture of cancer research. Our goal is to establish the cancer knowledge network to cover different aspects of cancer research, like the cancer prevention and cancer diagnosis, cancer therapy, and also the cancer monitoring. I think for the cancer prevention, it's very important to establish figure out the risk factors for the cancer development, so it can help us to develop the technology for early detection. And also in the area of precision medicine, it's very important to associate the cancer diagnosis, like the molecular classification to different kinds of clinical features, so we can guide the patients or the doctors to have a better therapy strategies. And also, because cancer is a very complicated disease, so we need to have some technology for the monitoring, like the MRD detection. So it's very important to develop a non-invasive detection technology, like the sequencing on the circulating tumor DNA. I think from cancer genomics to cancer medicine is the process that we first understand the cancer genomics to identify different kinds of biomarkers, and also not only analysis these data from single type of cancers, but also we can perform the more comprehensive analysis that at the level of pan cancer to associate much more biomarkers to figure out the common mutations or driver genes in different kinds of cancers, and also the specific targeted the mutated genes in the individual cancer types. And also based on the functional study that we can determine the specific mechanism of these molecules to the cancer development, to finally help us to set up this knowledge base to support the drug development, or also the detection or testing technology. So this is you can learn from the projects of ICDC and also the ICDC ARGO. And I think we can divide the omics research for clinical oncology into different parts, as I just mentioned. The first of all is the cancer genomics study. So we investigate the comprehensive characteristics of cancer genome using the multi-omics data to dissect the genetic mechanisms of tumor development. And it because of the heterogeneity of cancer, so we need a very high resolution technology like the single cell sequencing or spatial transcriptome sequencing to characterize the cancer heterogeneity and also the evolution dynamics during the cancer progression. And also we need to, based on the understanding of the biomarker or molecular patterns, we need to have some cohort study to associate these molecular patterns to some specific clinical features, so we can know more about how to make cancer precise prevention, diagnosis, and therapy and monitoring guidance. So in the site of real-world study, real-world clinical research. And finally, we based on this our knowledge of this cancer research, we can help us to develop some critical application technology, so we can perform this molecular testing for the patients to help them to have a personalized medicine to achieve better benefits from the clinical treatments. So I think thanks to the development of the next generation sequencing platform that opened the area of multi-omics investigation for the cancer research. So I think according to the practice of TCGA and ICDC, so we have achieved multiple omics data sets from diverse tumor types. So these kinds of data can enable the comprehensive analysis on numerous aspects of cancer genomics. But I think the challenge in fighting cancer, the common knowledge is that the inter and intratumoral heterogeneity is the core challenge in cancer treatment. And this is not just at the level of population, but also between different individuals. And even in the same tumor mass, the intratumoral heterogeneity that at the single cell level also is the very critical issues that we need to solve. So here I take an example of the liver cancer, that hepatocellular carcinoma. The liver cancer is one of the most common cancer types in the world, and especially in China. So we need to know more about the intratumoral heterogeneity, and also for the clinical application from the patients that at the advanced stage, that we need to provide the similarity between the cell-free-DNA and the tumor tissue, because for most of these patients at advanced or late stage, we can only collect the blood samples from them, and we need to identify the mutations that from the circulating tumor DNA that come from the cell-free DNA. So we need to understand that what we can detect from the cell-free DNA and how does this mutation can represent the real mutation in the tumor tissue. So we have this study that collaborates with the local hospital. So we created the tumor samples from the liver cancer patients, and notably we performed a multi-region sequencing, multi-regional tumor tissue sequencing to identify the intratumoral heterogeneity, and also we performed the comparison between of the mutation detection between the tumor tissue, the multiple tumor tissue, and the cell-free DNA. So first of all, we can find that there are many ubiquitous variants that are present in all regions, but also some heterogeneous variants that just share between parts of the regions, and also the private or we call it unique variant that exists only in one region. So you can see that the proportion of non-ubiquitous variants was defined as the level of the intratumoral heterogeneity. So you can see that different patients will display diverse patterns of the heterogeneity, and on average, about 35% of the mutations were non-ubiquitous. So we can see that ultra-deep sequencing that can help us to dissect more clearly of the heterogeneity, because only through the high depth of sequencing you can detect or identify the low frequency of mutations. So we can see that the heterogeneity is not only come from the SNVs, but also the copy number variation can represent the heterogeneity. So here from this analysis, we can see that the copy number variation differs more significantly among patients than across regions, but the CNV heterogeneity closely correlates with that of SNVs. So we can say that CNVs might share similar development process with the acquisition of SNVs. And from this phylogenetic analysis of the multi-region liver cancers, you can see that we identify some chance mutations that like the TP53, P10, that represent the clonal the mutation that may occur at a very early stage of the tumor development. But also we can identify some subclonal mutations that may represent the signals or patterns that for the tumor recurrence or metastasis, and also may responsible for the resistance to some specific therapy. So that may help us to understand the evolution of the cancer. And also, we as I just mentioned, we need to have some comparison of the genomic profiling that to determine the efficiency of cell-free DNA in liver cancer. So here you can see that when we compare the mutations that identified from the circulating tumor DNA and also the multi-region tissues, you can see that the mutations we identify from the cell-free DNA may represent more diversities from the tumor tissue, but because of the limitation of detection of low frequency mutations in cell-free DNA, so we need to combine both the multi-region tissue sequencing and also the cell-free DNA sequencing to have a more comprehensive mutational profiling of these individual patients. So another important part of the cancer research is the cancer immunogenomics and the immune microenvironment, because that the immunotherapy represents a very promising therapeutic strategies for cancer patients. So here also for the liver cancer, we set up this project to study the multifocal HCC. So the multifocal HCC can be classified into two groups. One is the hepatic metastasis, and another kind is the multi-century occurrence. So this represents different kinds of patient group. One is the late stage with metastasis, but another group is at the very early stage. So here you can see that distinct clonal architectures and also mutational signatures and genetic susceptibilities that between these two kinds of patients have been identified in this study. So you can see that for the patients with intratumoral metastasis, they will have more clonal mutations and subclonal mutations. But for the patients of the multi-century occurrence, that you can see a very different evolutional patterns. And here we also evaluate the heterogeneity of immune infiltrations between these two groups. That you can see that the immune cell infiltration is also associated with the mutational profiles. That because through this transcriptome sequencing, we can evaluate the immune environment that the context that consists of different types of immune cells. So this is very important that to have a better understanding of this immune cell or immune microenvironment will help us to understand why some patients may have better response to immunotherapy. But the limitation of the sequencing technology that we can only evaluate the proportion of different kinds of immune cells, but we need to have some special technology to figure out the spatial distribution and density of immune cells. So here we perform the multiplex immunostaining to assess this pattern. So I think maybe in combined with the sequencing technology will help us to have a more comprehensive profiling of this microenvironment. And also, you can see that we predict the new antigens from the mutation identification. So this is very important that we determine that 35% of predicted new antigens were able to invoke significant specific T cell expansion with the production of SN and gamma. And this is very meaningful because we can apply these personalized new antigens for to help to promote the benefits of immunotherapy, because we can develop the vaccines that target the personal new antigens for each patient, and may have better benefit from the help the patients to have better benefits from the immunotherapy. But as I just mentioned, we need to have higher resolution technology to help us to figure out the spatial distribution of the cells or the gene expression. So here, BGI have developed a spatial and temporal enhanced resolution omics sequencing approaches we call the stereo sequencing. So here, to get integrated with our sequencing platform, the DMV pattern chip, so we can have this in situ the mRNA capture and sequencing for the tissues. So here we take an example of the mouse embryo development at different time points. So we can very clearly see that we can identify the molecular patterns or the gene expression patterns from different organs. So here is I think here is the power of the spatial sequencing to help us the distribution of different molecular activation in different regions of the tissue. And when we apply this technology to tumor, you can see that, for example, this is also a liver cancer tissue. So you can identify the different patterns in the regions of a tumor and left, and also the adjacent normal tissues at right. And you can see that the interface, there are some enrichment of the immune cells that may represent the process of the cancer immunology. So here we propose that the subcellular resolution and panoramic field of view are crucial for spatially resolved technologies. And here, when we apply this technology, you can have the spatial transcriptome patterns that reveals the profiling characteristics of the tumor microenvironment. And also, I think finally, I think that based on our technology and the knowledge base we have established from this omics data, we can establish this pipeline for the oncological gene testing for clinical practice. That also not only for the mutations or biomarker testing, but also using the spatial technology, we can achieve the so-called digital pathology to help the doctors or the patients. They can have a better understanding or comprehensive or complete understanding of the profiling, the molecular profiling of the cancer tissue. So to identify some specific biomarkers or mutations or molecules that have a strong association with the clinical features to make better strategies for the diagnosis and therapy. So take it together so we can see the workflow for continuous discovery and translation, the study in cancer research. So the first part is the exploratory research that we can combine the omics data from the next generation sequencing with the clinical features to make the significant associations to identify some specific biomarker or mechanisms that after the big cohort validation, that both on the wet lab functional study and also the prospective clinical trials to achieve some conclusion that can support both the development of the diagnosis or testing the testing for diagnosis or therapy guidance, and also the pharmacy development. But the important thing is that I always think we need to have some close cooperations between the government, healthcare system, hospital, academic institution, and also the industry that to work together to share all of our data and technology to achieve a more comprehensive workflow to help the cancer patients to achieve better benefit from the diagnosis and also the therapy. So that's what I would like to share with you today. And any questions is welcome. Thank you.